A GCPR signaling pathway is a cell communication process where a molecule binds to a G protein coupled receptor, triggering a cascade of intracellular signals that regulate cellular responses. In one example, epinephrine binds to a GCPR on the cell surface. The receptor activates G proteins inside of the cell. The G proteins then trigger adenylene cyclase, an enzyme which produces cyclic AMP from ATP, abbreviated as CAMP. Cyclic AMP activates protein kinase A, which activates phosphorylase kinase, which then activates glycogen phosphorylase. Glycogen phosphorylase breaks glycogen into glucose 1-phosphate. The glucose is used for energy in muscles. A gated ion channel signaling pathway is a process where a signal triggers the opening or closing of ion channels in the cell membrane, allowing specific ions to flow in or out, which alters a cell's activity. In one example, capsaicin activates the TRPV1 protein channel in the cell membrane, which opens up an ion channel to allow calcium ions into the cell. This influx of calcium ions leads to the activation of the enzyme calcineurin. Calcineurin dephosphorylates the transcription factor NFAT2 found in the cytoplasm. The dephosphorylated NFAT2 then moves into the nucleus, where it blocks the transcription of another protein, ATF3. ATF3, when active, prevents the cell cycle regulatory protein, PGOG, from inducing apoptosis and blocking cell division. A receptor tyrosine kinase, RTK signaling pathway, is a cellular communication process where extracellular ligand binding activates RTK, triggering phosphorylation cascades that regulate cell growth and division. In one example, an epidermal growth factor reaches the RTK receptor within the cell membrane. From there, RAF kinase is activated. MAPK kinase is what it is also known as. RAF phosphorylates and activates MEK and MAPK kinase, which in turn phosphorylates and activates ERK, extracellular signal regulated kinase. ERK enters into the nucleus and phosphorylates target proteins. In the nucleus, CMYCR is phosphorylated and undergoes the transcription of genes promoting cell growth and division. An intracellular signaling pathway is a cellular communication process where signals enter a cell and bind to specific receptors, triggering changes in gene expression or cellular function. In one example, the hormone aldosterone passes through the plasma membrane into the cell cytoplasm. The aldosterone binds to a receptor mineralocorticoid protein in the cytoplasm, activating it. The hormone receptor complex diffuses into the nucleus and binds to specific genes on the DNA. The bound protein acts as a transcription factor, stimulating the transcription of the gene into mRNA. The mRNA is translated into specific proteins.